Hello everyone, today I'm going to teach you about the neuromuscular junction. So the neuromuscular junction is the junction in between the neuron right there and the muscle cell right there. And it's the way that they communicate one to another. So you start out, so this is you, and you have a brain, and inside of your brain you have a whole bunch of cells called neurons, and when you have a thought like, oh, I'm going to move my hand, that signal has to be sent through a neuron down your spinal cord into your muscle. And right here, we have a picture of a motor neuron. And that is, here's the neuron, and then it comes down and it kind of spider webs onto your muscle cell, kind of like that, onto a few muscle cells. And here is a zoomed up version of that junction. So let's just start from the top. An action potential sweeps down the neuron, and that's a series of sodium voltage gated channels opening. So as more positive ions flood the neuron, more voltage gated sodium channels open, which causes more sodium to come inside of the cell, like so. And when the voltage gated sodium channel opens, it triggers the calcium voltage gated channel to open. This is calcium voltage gated channel. So calcium floods into the neuron, and it binds over here to a snare protein right there, to this golden thing, which is called a snare protein. Now, the snare protein pulls through. You see right here, you have these vesicles full of these red dots called acetylcholine. Acetylcholine is a neurotransmitter. And what happens is when your calcium comes inside, it allows the acetylcholine to bind onto the snare protein. And then it causes exocytosis, which looks like that. So exocytosis, so you have your phospholipid bilayer, and then here's a vesicle, and then it slowly becomes part of the vesicle like that. And then all of the things that were inside of here, they come out like so. Exocytosis. So the calcium binds onto the snare protein over here. Acetylcholine vesicle binds, and it requires calcium to bind, and that causes exocytosis of acetylcholine. Now, once acetylcholine is inside of the synaptic cleft, that's the name of this area right inside of here, right in between the neuron and the muscle, that is the synaptic cleft. And that is because that is where the synapse is happening. Synapse. Synapse just means the message is being transferred from one cell to another, from either a neuron to a neuron or a neuron to a muscle. So acetylcholine comes down here into the synaptic cleft, and these little red guys, Pac-Man, they come and chomp up the acetylcholine when it's through, and that right there, that's acetic acid, and this is choline. The choline gets recycled back into the cell, and the acetic acid, it goes to other parts of the body. Okay, so once acetylcholine is inside of this synaptic cleft right here, it comes and binds to a ligand-gated cationic channel. Ligand-gated cationic channel. And all that means, a ligand is a key. So it's key gated, it requires a key to open, and it lets in cations or positive ions. So once acetylcholine right here, acetylcholine is the key or the ligand. So when acetylcholine comes into the synaptic cleft, it binds onto the ligand-gated cationic channels, which, by the way, these type of ligand-gated cationic channels are known as nicotinic type 1 receptors. And so they open, and it lets in cations, in this case more sodium. So sodium is flooding into the muscle now. Okay, so that's part 1. Part 2 is down here in the muscle. So what happens is, as positives come into the cell, they need to be dispersed somehow to go deep into the muscle. And the way they do that is through these things called T-Tubules. Now, T-tubule, it's kind of like a donut. It's poked all the way through the muscle cell and it extends all the way down. So that way, you can have sodium-gated channels on either side, like that and like that. And this is still part of the muscle cell. This is still part of the muscle cell. They're both. This is only one little section of it, but the T-tubule extends all the way down through the muscle cell, so as to propagate the action potential more quickly to the middle. So as positive ions flood into the cell right here, then that causes these voltage-gated sodium channels to open, letting in more positive ions, and that propagates down the T-tubule until it opens a voltage-gated protein right here called DHP. So DHP, that's the darker green one. Here, let me draw that in a darker green color. DHP. So this is what DHP looks like when it's closed, and this was what it looks like when it's opened. So again, closed, open. So DHP is triggered by all of these positive ions coming into the cell. When DHP opens, that also opens RYR, which is this light lime green structure right there, RYR. And this is what it looks like when it's closed. So when DHP opens, it opens up ryanodine like so. And that allows calcium to flood out of the sarcoplasmic reticulum. So calcium is inside of your sarcoplasmic reticulum, it's being stored, and it comes out now. Comes out. Calcium binds onto troponin, that's this red line right here. These red dots right there and right there. So calcium comes and binds onto troponin, which removes tropomyosin, which is this green rope. And that exposes the myosin binding sites, which allows the cross-bridge formation to occur. Cross-bridge formation is when these yellow fibers back here, which are myosin fibers, the heads of them, they will actually pull the blue actin inwards, like so, like that. And that is what we call a muscle contraction. So as these get pulled inward like that, the muscle is contracting. So in order to end the contraction, calcium, which is right here, it needs to go back into the sarcoplasmic reticulum via a pump. The pump is called SERCA, S-E-R-C-A, the SERCA pump. And if the SERCA pump isn't working, then calcium can't go back inside, so you will be in a perpetual state of contraction, which we do not want. Okay, so just to run over things really quickly one more time, start from the top. An action potential sweeps down the neuron, opens a voltage-gated sodium channel. Sodium floods into the cell, which opens a voltage-gated calcium channel. Calcium floods into the cell, binds to a snare protein, causing exocytosis of acetylcholine. Acetylcholine binds to a ligand-gated cation channel, which allows cations to flow into the cell. And then let's move down a bit. So now an action potential propagates down a T-tubule, which causes DHP to open, which causes ryanodine to open, or RYR. Calcium floods out of the sarcoplasmic reticulum, binds to troponin, which moves tropomyosin, exposing the myosin binding sites on the actin. And then you have cross-bridge formation, which consists of binding, power stroke, release, and recovery. You do that for a bit until the muscle is contracted. Once it's ready to be done, then calcium will go back into the endoplasmic reticulum via the SERCA pump. And when acetylcholine is finished, then this little enzyme right here, Pac-Man, acetylcholine esterase, will chomp it up and spit it out as acetic acid and choline, which will be recycled. By the way, if your acetylcholine esterase doesn't work, what will happen? That's the question for the day. Okay, hopefully that was a helpful video, and yeah, good luck.